This hearing will come to order, and without objection, the chair is authorized to declare recess at any time. Let me say good morning and welcome to our witnesses. Today's hearing on vaccine science and innovation. Smallpox once plagued the world's population, killing approximately 300 million people in the 20th century alone. Smallpox is the only human disease to be eradicated, thanks to the development of the vaccine. Another devastating disease, polio, had just 33 cases reported worldwide in 2018, compared to 350,000 cases in 1988. Every day, vaccines are saving lives, especially the lives of children and other vulnerable populations. There's no such thing as healthy skepticism when it comes to vaccines. Unfortunately, there is a well-funded disinformation campaign targeting the public and weakening public health laws. School vaccination requirements have been commonplace in the U.S. for generations, and exemptions were granted only for legitimate medical reasons. However, in my home state of Texas, the number of unvaccinated children has spiked since 2003, when the Texas legislature expanded the exemptions to include non-medical reasons. The number of exemptions rose from 2,000 in the year of 2003 to 57,000 last year. We're seeing this replayed across the country, and innocent children are falling ill. Health officials have confirmed 21 measles cases in Texas this year, and 1,261 nationwide, 61 of which led to serious complications. As the first nurse elected to Congress, I have been dedicated to the improvement of public health my entire career. The Science Committee may not have jurisdiction over the health and human service agencies, but we have long had a role in supporting improved public health through good science. This morning, we will explore the science and innovation challenges for vaccine development through the lens of influenza. For the healthiest among us, the flu just lays us out for several days with no lasting side effects. However, for the very young, the elderly, pregnant women, and other vulnerable groups, the flu can be deadly. The Centers for Disease Control recorded an estimated 48.8 million illnesses and 79,000 deaths during the 2017-2018 flu season. Approximately 600 of those deaths were children. Each year, influenza vaccine production begins with the collection and analysis of data many months before the beginning of the flu season. The challenge with influenza is that the viruses change constantly, and by the time the flu season begins, the vaccine may not fully match the circulating viruses. Scientists are working to develop viable and more effective alternatives to the current egg-based vaccine, as well as a universal vaccine that will not require annual update. Yet another scientific challenge for influenza and many other infectious diseases that are incomplete is incomplete data and antiquated data systems. Through modernization of data systems and data analytic tools across the federal and state levels, we will be able to accelerate vaccine research and development for many diseases. We have two expert panels that will help us understand the full cycle from basic research to vaccine development, production, and deployment, and surveillance. The witnesses will also describe the role of federal agencies, state agencies, and the private sector, including the partnerships among all of the stakeholders. I want to extend my warm welcome to all of you this morning, and I want to thank the Vice Chair, Dr. Barrett, for his leadership on this issue. I look forward to today's discussion. I might say that I have a markup in another committee, so I will have to leave before we get through all of the deliberations. The chair now will recognize Mr. Lucas for an opening statement.
Good morning, Chairwoman Johnson. I would like to thank you and Vice Chairman Barrett for holding this hearing, especially given that we are in the middle of flu season. In the United States, nearly 1 million individuals are hospitalized for the flu every year, including more than 48,000 children. In Oklahoma, since the 2019 flu season began on September 1, there's been at least 1 death and 73 hospitalizations from the flu. However, these numbers would be far worse if we did not have vaccines. Vaccination is by far the best flu prevention measure we can have today. It's easy to forget that a little over 100 years ago, the world faced one of the deadliest pandemics in history, the 1918 H1N1 epidemic, also known as Spanish flu. It killed an estimated 50 million people worldwide, including roughly 675,000 people in the United States. Medical technology and countermeasures at the time were limited to isolation and quarantine. Influenza vaccines did not exist, and antibiotics had not been fully developed yet. Thankfully, due to basic research, advancements were made both in treatment and prevention of the flu. The development of vaccines has played an important role in reducing and eliminating deadly disease. I can still recall my father's stories about how late summer and fall were a terrifying time as a child because of the threat of polio during those seasons. Lucky for me, I did not have to experience this fear because of the first polio vaccine being available in the United States in 1955. And thanks to widespread vaccination, polio has been nearly eradicated in the United States, with just 33 cases reported in 2018. However, polio remains a threat in some countries. With the world becoming more connected through modern transportation, it only takes 1 traveler with polio to bring the disease into the United States. And as I'm sure we'll hear this morning from our witnesses, the best way to keep the United States polio-free is to maintain high immunity through vaccination. Considerable advancements have been made in health technology, disease surveillance, medical care, medicines, drugs, vaccines, and pandemic planning. While significant progress has been made, gaps remain, and a severe pandemic could still be devastating to the global population. As the human population has grown, so has the livestock, swine, and poultry populations to feed them. This expanded number of hosts provides increased opportunities for unique viruses from birds, cattle, and pigs to spread, evolve, and infect people. As a member of the House Agriculture Committee, I supported the creation of the National Animal Vaccine and Veterinarian Countermeasures Bank, which was included in the last Farm Bill. This vaccine bank will maintain a sufficient quantity of animal vaccines and other countermeasures to provide a rapid response to an animal disease outbreak. If an outbreak were to occur and we were not prepared, our entire agricultural sector would suffer immense losses, causing long-term harm to the economic viability of the United States livestock, poultry, and swine production, not to mention the damage to human health. I look forward to hearing from our witnesses today about the current state of our stockpiles of human health vaccines to provide the capacity for rapid responses to emergency situations. I particularly look forward to hearing how BARDA influenza vaccine manufacturing infrastructure is supporting the public-private partnerships with domestic vaccine manufacturers to increase preparedness levels and response capacities for potential pandemic flu events in the United States. Lastly, I would just like to say how pleased I am to see the President's recent executive order to address modernizing flu vaccines. The executive order recognizes that influenza as a public health and national security priority, with the potential to inflict harm on the United States through large-scale illness and death. Most importantly, it establishes a national task force to explore alternative vaccine production methods and new technologies, including a plan to accelerate the development of universal flu vaccine. I look forward to seeing what recommendations come from the task force. I would again like to thank Chairman Johnson and Vice Chair Barrett for holding this hearing. I would also like to thank both witness panels for taking the time to share your expertise, your insights with us this morning. And I yield back the balance of my time, Madam Chair.
Thank you very much. If there are members who wish to submit additional opening statements, your statements will be added to the record at this point. At this time, I will introduce our witnesses. Our first witness on the panel is Dr. Daniel Jernigan. Dr. Jernigan is the Director of Influenza Division for the National Center for Immunization and Respiratory Disease at CDC. Dr. Jernigan is responsible for oversight and direction of a broad scientific program to improve detection, prevention, treatment, and response to seasonal, novel, and pandemic influenza. The Influenza Division is responsible for national and global surveillance of influenza and serves as a World Health Organization collaborating center for the surveillance, epidemiology, and control of influenza. Dr. Jernigan entered the CDC in 1994 and is a retired captain of the U.S. Public Health Service and was a recipient of the 2019 Service to America Medal. The next witness on this panel is Dr. Anthony Fauci. Dr. Fauci is the director of the National Institutes of Allergy and Infectious Diseases, a position he's held since 1984. He oversees an extensive research portfolio of basic and applied research to prevent, diagnose, and treat established infectious diseases such as HIV/AIDS, respiratory infections, diarrhea diseases, tuberculosis, and malaria, as well as emerging diseases such as Ebola and Zika. He also supports research on the transplantation and immune-related illnesses, including the anti-immune disorders, asthma, and allergies. He has advised 6 presidents on HIV/AIDS and many other domestic and global health issues. He was one of the principal architects of the President's Emergency Plan for AIDS Relief, a program that has saved millions of lives throughout the developing world. As our witnesses should know, you will each have 5 minutes for your spoken testimony. Your written testimony will be included in the record for the hearing. When you've completed your spoken testimony, we will begin with questions. Each member will have 5 minutes to question the panel. We will start with Dr. Jernigan.
Well, thank you very much. Good morning, Chairwoman Johnson, Ranking Member Lucas, and distinguished members of the committee. I am Dr. Dan Jernigan, Director of the Influenza Division at the Centers for Disease Control and Prevention. I want to thank the committee for the opportunity to discuss CDC's work supporting vaccine innovations to improve prevention of influenza. Each year, influenza causes a significant health burden in the United States, with many millions of Americans becoming ill, hundreds of thousands requiring hospitalization, and tens of thousands dying. Influenza viruses are constantly changing, requiring us to update the vaccine components every year. Sometimes these changes can be sudden and significant, resulting in flu strains that can lead to devastating pandemics. Hospitalization and death can happen in any flu season, and each year flu vaccination prevents millions of illnesses and thousands of severe and sometimes tragic outcomes. Influenza vaccines are very safe, and they remain the single best way for people to fight the flu. Despite the significant benefits, the effectiveness of the flu vaccine and the numbers of Americans being vaccinated are not optimal. We at CDC are working with NIH and other federal and state government partners and with the private sector to use cutting-edge science to make influenza vaccines better. The long-lasting, broadly protective universal vaccines that Dr. Fauci will talk about are the ultimate goal for flu prevention. However, these vaccines are still years away. In the nearer term, we can save millions of Americans from the flu by making incremental improvements to vaccines that can be produced using already available production platforms and by getting more Americans vaccinated each flu season. CDC has a central role in every part of the seasonal influenza vaccine development and administration cycle, including continuous virus tracking around the globe, preparation of vaccine viruses, purchasing 10% of flu vaccines used in the United States, and monitoring vaccine coverage, safety, and effectiveness. To improve flu vaccines, CDC has implemented innovations throughout the vaccine life cycle. CDC has invested in and collaborated with every state public health department on flu surveillance. This investment has resulted in automated, real-time electronic laboratory reporting of influenza test results to CDC using cloud-based messaging. CDC has transformed flu virus surveillance by using next-generation genomic sequencing to characterize all influenza specimens received at CDC. This means we can identify and track viruses much more quickly and accurately, leading to more timely selection of candidate vaccine viruses and earlier detection of viruses with pandemic potential. Genomic sequencing equipment, which once filled a room, now fits in the palm of your hand. We now have a mobile mini lab that can be taken on the plane as a carry-on and set up almost anywhere in the world, including rural resource-constrained settings. CDC has implemented innovations for supporting newer vaccines by developing candidate vaccine viruses for the cell-based vaccine and by providing genomic sequences used to make the recombinant protein vaccine. Both of these newer vaccines have the potential to be manufactured more quickly and may be more effective than traditional vaccines that are grown in eggs. CDC now also routinely generates vaccine viruses using a technique called reverse genetics. This allows us to build a vaccine in a matter of days or weeks, much faster than traditional methods, making the U.S. more prepared to respond quickly to a pandemic. CDC was the first to establish a national system for the routine monitoring of influenza vaccine effectiveness, and that vaccine effectiveness network provides critical information for manufacturers and researchers in developing enhanced vaccines by collecting more specific data about how well the vaccine works each season. Recently, we have expanded the network and are planning to add new immunity testing and conduct more studies to better evaluate vaccine effectiveness. Finally, a major component of improving influenza vaccine impact is getting more people vaccinated. Fewer than half of adults in the U.S. receive their influenza vaccines. And despite all of our successes and our global leadership in influenza detection and prevention, there is still more we need to be able to do. Each of the topics I mentioned today, from working with domestic public health partners to track and characterize viruses, to developing vaccine candidates and studying vaccine effectiveness, will benefit from investments in generating more precise and timely data. I believe we can harness this data to make vaccines work better. I want to close today by reminding you all to make sure that you and your families are vaccinated before the holiday travel begins. And thank you for the opportunity to talk about CDC's influenza work, and I look forward to your questions. Thanks.
Thank you, Dr. Jernigan. Dr. Fauci.
Thank you very much, Madam Chairman. Members of the committee, thank you for giving me the opportunity to testify before you today. I am Dr. Anthony Fauci, the Director of the National Institute of Allergy and Infectious Diseases at the NIH, and I'm going to talk to you over the next couple of minutes about the NIH's efforts to improve the influenza vaccines and to ultimately develop a universal flu vaccine. As shown on this slide, although, as Dr. Jernigan had mentioned, it's very important to get vaccinated because even if a vaccine is not 100% effective or even 50% effective, the benefit to the individual to get vaccinated and to the community is profound. However, we can do better because seasonal influenza vaccines are not consistently optimally effective. In addition, we know through history that pandemics occur, but we usually are too late in our response, as we were in the 2009 H1N1. And finally, we spend considerable time, what I call, chasing after pandemics, as we had with the H5N1 and H7N1, in which we made significant investments. We needed to do that, but those pandemics never occurred. This slide shows a journal, the Journal of Infectious Diseases, containing a number of papers in which my colleague and I gave the introduction, emphasizing the point that I just made, that although influenza vaccines are good and important and should be utilized, we can do better. By doing better, we need to improve the seasonal influenza vaccines, which would lead to a better capability to respond to pandemic influenza, which ultimately will get us to the goal that we'll speak about over the next minute or two, and that is the development of a universal influenza vaccine. In the summer of 2017, we brought a group together to develop a plan which we published in 2018 for the strategic plan and the research agenda to mobilize scientists throughout the country and the world to develop a universal flu vaccine. So let me explain what we mean by a universal flu vaccine. This is somewhat of a complicated slide, but it really does make the point. We will not get a universal flu vaccine overnight. I use the word iterative, which means it will be a stepwise process in which we go from improvement, the broad capability of responding to a particular type of a strain versus the ability to respond to all strains. Note on the lower left-hand part of the slide, it is divided into two major groups of influenza, Group 1 and Group 2. On the right-hand part of the slide, the tip of that triangle is what we do today. We make a vaccine for this season that's highly specific to the strains that are circulating this season. However, those strains change, they mutate, they drift. What we want to do is go to the next step, is to make a vaccine that would cover all the H3N2s or all the H1N1s. And then the next step would be to get one that would do all the Group 1s and all the Group 2s until ultimately we have a universal vaccine that essentially covers all of these. We're going to do that with new technologies. As you're well aware, we currently have a technique of growing the virus in eggs to develop a vaccine. Although that's tried and true and time-honored, it's inefficient and has many areas of going wrong. So we're using new platforms, as shown here on this slide, such as recombinant proteins, viral vectors, nanoparticles, and others. This is a blow-up of the influenza virus, and to the right is an important protein called the hemagglutinin. It is important to note that the hemagglutinin has two components, what we call a head and a stem. The head is the part that the body makes an immune response against. However, it mutates often, changes, leading to the ineffectiveness. However, the dark blue is the stem, which doesn't change much at all. So the strategy now, one of several strategies, is to develop a vaccine in which you cut off that head, as shown there, take the stem, and put it on a nanoparticle, which is highly immunogenic, which will ultimately serve as the vaccine. So if I could show you this, this is a 4 million times blow-up of what the first universal flu vaccine would look like. And these dark blue areas are the stems. We have started a phase 1 trial, as shown here, in the spring of this year. It will end at the end of this year. And then next year, we will do a Group 2 universal flu vaccine. So as the President said in the executive order, the purpose of what we're doing is to go ahead and improve little by little till we get vaccines to protect us in the most efficient way possible. Thank you.
Thank you, Dr. Fauci. At this point, we will begin our first rounds of questions, and the chair will recognize herself for 5 minutes. Dr. Jernigan, as you well know, there is a well-funded disinformation campaign sowing confusion and fear in the public. This campaign carefully targets and preys on different populations with different belief systems. Innocent children are falling ill today with diseases we once thought were eradicated in the U.S. Young women are unnecessarily being put at increased risk for cancer. And these anti-science forces are creating a major challenge in future vaccination efforts. How big of a role does social media play in this resurgence, and how can we overcome these tactics? And what is CDC doing specifically to combat these efforts?
Yeah, so certainly at CDC, we want to do everything we can to get more people vaccinated. We know with influenza that only about half of Americans actually get vaccine. Another half still need to get vaccinated. A lot of the reasons why they don't get vaccinated is for influenza is because of worried about the effectiveness of the vaccine. So with regard to our discussion here, improving the effectiveness of the flu vaccine certainly would actually, I think, get more people to be vaccinated. Your question is really around the role of misinformation and social media in that, in participating in that. We do think that there is a lot of information out there. Parents have lots of different places that they can get information, and a lot of times they don't know which of it is science-based, which of it is evidence-based, et cetera. So I think at CDC, our plan is really to try and strengthen public trust in vaccines by truly trying to get people to be more confident in the vaccines, getting the information out there about how effective they are. And that really comes down to three things: protecting the community, helping to understand the differences in these different pockets, these different communities, what makes them not have as much confidence in vaccine as they should, and identify and develop materials that we can use for those specific communities, reaching out to key opinion leaders within those communities. A second thing would be to empower the parents. That is, get with the very young parents when they first have children, or with pregnant women, get them the right information that they could understand better about the benefits of vaccine and understand why it is so important to get vaccinated, and work with clinicians so that they have the tools to talk with those family members as well. And then finally, to stop the myths as much as possible. And so we do that, I think, by providing the scientific-based, evidence-based information that's out there, it's on our website, and then working to make sure that that can be reused on multiple different platforms so that people can get that science-based or evidence-based information.
Thank you very much. Dr. Fauci, would you like to comment on that?
Yeah, just to underscore what Dr. Jernigan said, if you do a survey and find out what is the most important reason why people don't get vaccinated for influenza, and it's because of the so-called misperception that it really doesn't work. And I think we need to emphasize that even though it isn't 100% effective, even a modestly effective vaccine will prevent you from getting infected, will prevent individuals, particularly those who are susceptible to complications, will prevent them from getting hospitalized, and may ultimately save their lives. So this perception that the vaccine doesn't work, really we need to put that aside, because everyone, as Dr. Jernigan said, should get vaccinated.
Thank you very much. I'm going to ask Mr. Lucas to ask his.
Thank you, Madam Chair. Dr. Jernigan, in Oklahoma, the State Department of Health has reported that influenza has already claimed 1 life and hospitalized 70 others. Continuing on the comments that you and Dr. Fauci have made, when I look my constituents in the eye back home to stress the importance of getting vaccinated and to prevent the hospitalizations, death, could you expand on that? You're in a town meeting with me, you're looking my neighbors in the eye, this is rural Oklahoma, you're talking about things that are to the point.
Well, certainly we know the burden of influenza is very high. That is, the illnesses and deaths that occur because of influenza. There are tens of millions of cases every year, hundreds of thousands of hospitalizations, and tens of thousands of deaths that occur every year. We know that with the vaccine that we have, you can prevent thousands of deaths every year and tens of thousands of hospitalizations. It's important to get vaccinated, not just for yourself, but because it also helps protect the community around you. There are a number of benefits that the vaccine has. It prevents you from getting sick. It reduces you from having to be hospitalized with flu. For people with underlying chronic diseases, it's actually like a prevention tool. It's like something you should take every year because it can keep you from getting a second heart attack. So people with underlying conditions, it helps them as well. It protects pregnant women and their babies so that those that are born but not 6 months and eligible for vaccine, getting the pregnant mother vaccinated actually helps the baby during that period of time before they can get vaccinated. There's data that shows that it's life-saving in children. You can actually reduce the chance of death with influenza by 65%. So there are a number of things that are important about it. Even if it's not 100% effective, like Dr. Fauci mentioned, it can reduce the severity of illness that you have during the flu season if you were to get infected.
Dr. Fauci, my background is in agriculture, and in that world, of course, we have the robust National Veterinary Stockpile, which is prepared to provide farmers and ranchers with countermeasures against damaging animal diseases such as avian influenza and swine flu within 24 hours. Could you speak to the current state of the human vaccine stockpile management and what we could do to better prepare to address potential pandemic emergencies?
Sure. I would love to do that, except that the CDC is the one who's responsible for the stockpile, so I'll pass that to Dr. Jernigan.
I'll flip over to you then, Doc.
not to keep passing this, but actually BARDA is the one that manages manages the vaccine.
With the great
stockpile.
insight that both of you have,
Yeah, we can-
enlighten us in what's going on so we can reassure the folks back home
Sure.
we're paying attention. That is, you and your entities are taking care of their best interest.
Absolutely. So I think in terms of what we do at CDC, we monitor influenza around the globe, especially the avian influenza and the swine influenza viruses that are emerging around the globe. We do that through 143 laboratories where we detect those. We take that information and use it in a thing called the Influenza Risk Assessment Tool, or the IRAT. You can actually get on your browser and put in IRAT CDC and see a graph of where we have ranked these different concerning potential pandemic viruses in that graph. With that information, we work with the rest of the interagency to determine which of those should be made into vaccine candidates, which of them should actually be made into vaccines and stockpiled, which ones should undergo trials. And so with that, we have made decisions about things to put into that stockpile so that the U.S. is prepared. Many of those vaccines, say for instance the H5N1 vaccine, it's in the vaccine stockpile. It may be enough to vaccinate first responders and a few small risk groups. However, these viruses continue to change. And so it's actually very important for us to find new vaccine technologies so that the vaccine stockpile isn't something that just has to keep getting more and more vaccines put into it, but rather upstream we have fast technologies that will be able to make vaccines quickly. And then ultimately, once there is a universal vaccine, that may be the best thing for us to prevent pandemics, is to have that available.
In my final moments before I yield back, I alluded in my opening statement to my father's observations in the 40s and 50s prior to the development of the polio vaccine in 55, how the outbreaks kept seemingly getting worse and worse, and the sheer terror that it brought in the communities in that late summer season and early fall. My generation was not alive for that, did not experience that, but it was truly terrifying. My first off-farm job when I was 14 was mowing a little country cemetery. And I had an great aunt who was the family historian. And I remember asking her why, in one section of the cemetery, why are all these babies buried? Why are all these young women buried? She said, look at the tombstones. They say 1918 and 1919. The Spanish flu took them all, and brought, even in rural Oklahoma, society to a grinding halt for weeks and weeks as this passed through. My generation, having not experienced any of that, sometimes doesn't necessarily understand what the potential downside is and why you gentlemen and all of your colleagues work so hard. And thank you, Dr. Barrett, for giving us this opportunity to focus on these issues. And with that, I yield back.
Thank you to the ranking member. Also, thank you to Chairwoman Johnson for allowing me to be a doctor today. And I think there are a couple hearings happening on the Hill today. I think this is the most important hearing that's taken place actually today, and I think that's why all the cameras are out in out in Longworth. Just thinking about it to both Dr. Fauci and Dr. Jernigan, my home district and my home state senator is Dr. Richard Pan, a colleague of mine, and we're on the front lines of trying to combat some of the disinformation that is out there. And I just want to run through a couple quick yes/no questions. Is there any scientific evidence that vaccines lead to increased risk of autism, Dr. Fauci?
Absolutely not.
Dr. Jernigan? When I was practicing, I would talk to some of my patients. They would often come back at me and say, well, I don't want to get the flu vaccine because I had it before and it causes the flu. Dr. Fauci, is there any evidence that the flu vaccine causes the flu?
The flu vaccine does not cause the flu.
Dr. Jernigan?
I agree, the flu vaccine does not cause the flu.
Great. The whole point of science is to pursue the truth, and I think it's important for us to dispel some of these myths. There are legitimate reasons for a small cohort of individuals, if they have allergies to eggs, et cetera, to opt out of the vaccine. But one of the most important things about why it is important, let's use measles as an example, to vaccinate a large population of folks, is the concept of herd immunity. And I think it's important for the public to understand that particular concept. Dr. Fauci or Dr. Jernigan, whoever, would you,
It's a very important concept, not only for flu, but I mean, our recent unfortunate experience that we had in this country, particularly in New York City, in the Williamsburg section, was a classic example of what happens when the umbrella of herd immunity goes down below a certain level. Because you had a community in which the level of vaccination was somewhere between 70 and 80%. For measles, you need somewhere between 91 and 93 or more % of the community, so that when someone inevitably comes in from the outside or someone travels and brings back measles, if the community isn't protected by that herd immunity, you get the very unfortunate situation which we saw in the Williamsburg section of Brooklyn.
what are current measles vaccination rates in America?
It's over 90%.
Okay, so we want to keep that.
Right.
And measles was a disease that, for the most part, we had eradicated in America, and now we're starting to see that, the incidents starting to pop up again. I guess for Dr. Jernigan, I'll often hear individuals say, well, we don't really need these vaccines or the flu vaccine because we haven't had a pandemic like the Spanish flu for 100 years. Can you talk a little bit about why we've been so lucky?
Yeah, so I think with the pandemic influenza, this is a situation where the flu viruses that are actually circulating in animals can actually mix with those flu viruses that are in humans. And when they do that, they can they share their genes and can create a flu virus that has not been seen before. That means that it can spread very quickly through the community, and often it can cause severe deaths and illnesses and hospitalizations. The 1918, like was mentioned, was one of the worst. That one clearly caused at least probably 675,000 deaths in the United States. We've had 3 other pandemics in the last 100 years. Those were with changes in the vaccine that were not as bad. We at CDC have looked at the 1918 virus and found that there are particular changes in that virus that really made it severe. So there's nothing preventing that from happening again. So for us, it's important to maintain the vigilance so that we can see what's happening, maintain the ability to have vaccine available quickly so that we can get it, and be able to prevent influenza and severe influenza if we were to have another pandemic.
And in today's interconnected world where people move across boundaries, having 2 big oceans are not necessarily protective for us. Dr. Fauci, you and I had the opportunity to work together around the 2014 Ebola outbreak in West Africa. Can you talk a little bit about the evolution and development of an Ebola vaccine and how that's helped us, in the 2017 outbreak in Western Congo and given us the ability to better manage Ebola?
Well, back in the 2014 to 16 outbreak in West Africa of Ebola, during that period of time, we, together with a variety of other agencies, including the CDC and other international agencies, began the testing of a vaccine called VSV, which now is ultimately made by the company Merck. So at that time, we did phase 1 studies right here in the United States. We did it at the NIH on our campus. Some were done in Europe, and then we did it in West Africa. We advanced to phase 2. The CDC did a study in Sierra Leone. We did one in Liberia, and then ultimately it was shown in a ring vaccination study in Guinea to actually be effective in preventing infection, particularly those who were exposed. That vaccine has now been used in the Democratic Republic of the Congo, and over 245,000 doses have been given in a contact ring vaccination approach. It is very clear that if, in fact, we didn't have that vaccine, we would be in a much worse situation than we found ourselves in the Democratic Republic of the Congo. And as you well know from the reports coming out from the CDC, the number of cases per week of Ebola have gone down and down and down. We're not through with it yet. It's still there. But the vaccine has played a major role in being able to prevent the explosion that we saw in West Africa.
Well, Dr. Fauci, Dr. Jernigan, thank you for your service to our country. And just in closing, vaccines are safe, vaccines are effective, and vaccines save lives. So with that, Mr. Posey.
Thank you. I'm grateful to the chair for holding this hearing. Flu shots can play a very important role in protecting the public from the flu and reducing its spread. I want to focus my comments on a 90-year-old policy which should have ended decades ago. Why do we still have mercury in millions of flu vaccines that are given to infants, toddlers, and pregnant women? In July 1999, the Public Health Service, the American Academy of Pediatrics, and vaccine manufacturers issued a joint statement agreeing that thimerosal-containing vaccines should be removed as soon as possible. At this point, I have a number of documents that I would like to include in the record by unanimous consent. First is a bibliography of studies raising safety concerns about thimerosal, which is vaccine mercury. Second, a report from the Children's Health Defense outlining some of the misconceptions about mercury in vaccines, clearing up some misconceptions. Third, a 1999 joint statement of the American Academy of Pediatrics and the U.S. Public Health Service, calling for the immediate removal of mercury from all vaccines. In 2004, the Institute of Medicine recommended removing mercury from all vaccines administered to pregnant women and children. By 2003, mercury was removed from vaccines in the United States. Yet, a year later, the CDC recommended the flu vaccine for children 6 months to 36 months of age, but refused to state a preference for mercury-free vaccines. Thus reintroducing mercury to the childhood vaccine schedule. In 2006, California passed a law banning mercury-containing flu vaccines for pregnant women and children under 3. In 2009, much to the credit of Chairwoman Johnson, a bill was introduced banning mercury from power plants, and I think what she said then is even more pertinent to vaccinations, that mercury is a neurotoxin. Even at low levels, mercury can have an adverse health effect, particularly on women of childbearing age and on developing fetuses. Dr. Fauci, you worked with my predecessor, Dr. Dave Weldon, and I reviewed your testimony from October the 5th, 2004. That hearing was on removing mercury from flu vaccines. During that hearing, CDC Director Gerberding, the FDA's Dr. Egan, and you all agreed, and you stated repeatedly, we are moving rapidly to thimerosal-free vaccines. You also said the better part of valor is that if you can move to a vaccine preparation that is absolutely risk-free with regard to mercury, then you should do it. The public concerns are still there. Mercury is, in fact, a neurotoxin. Babies, unborn and newborn, are at a critical stage of neurodevelopment. The one change is what flu vaccine became a recommended shot. Manufacturers were automatically protected from all liability and accountability via lawsuits. Now they have no incentive to remove mercury. I read over the flu vaccine package inserts for flu vaccine, and each one says it has not been tested for safety in pregnant women. Common sense said that we should err on the side of safety. Dr. Fauci, you testified to that 15 years ago. The failure to completely remove mercury feeds the fear and takes a back seat to saving a few bucks each shot. What steps are being taken by you as a leader in the public health community to move, quote, rapidly to mercury-free vaccines, close quote, or is it no longer a priority, and when can we expect it to be completed?
I don't think I can answer directly the question of when it will be completed. Just getting back to the discussions that we had years ago in the committee, I said then, and I would say it again, that the optimal situation would be to have thimerosal-free vaccines. Mostly, as I mentioned at that hearing, which you didn't say, was mostly for the peace of mind of people, but the scientific evidence that is a harmful amount of this material in the vaccine does not indicate that. The issue with the thimerosal, and I'll let Dr. Jernigan also comment on that with regard to the CDC, is that it is in very, very few vaccines and only in multi-dose components. And the multi-dose component, the balance of the risk of getting a contamination of a bacteria which we know can occur if you don't put something like the thimerosal into the vaccine, versus the risk of a deleterious effect of thimerosal, which is really ethyl mercury and not methyl mercury, clearly balances the favor of making sure you protect from infection the multi-dose vial. So, but Dan, maybe you could amplify that a bit.
Yeah, I think it's important to know that CDC is committed to assuring that vaccines in the United States are safe. Currently, this year, there's projected to be 169 million doses of influenza vaccine, and we understand that only about 15% of that is the thimerosal-containing multi-dose vials. So those that would like to have a thimerosal-free vaccine, actually, there are the vast majority of vaccine that is available are the pre-filled syringes, the single-dose vials.
My time has expired. Thank you.
Thank you. Before I recognize Mr. McNerney, just a quick question. Mr. Posey raised a couple issues and maybe just yes/no answers. Is the flu vaccine safe for pregnant women?
Yes.
Yes, absolutely.
Is the flu vaccine safe for infants and children?
Yes.
Yes.
Great. With that, I'd like to recognize the gentleman from California, Mr. McNerney.
The neighbor from California. Thank you, Chairman. I thank the witnesses this morning. Dr. Fauci, how can computational data scientists partner better with microbiologists to accelerate the research?
Well, I mean, computational biology is a discipline that essentially impacts on virtually all of the biological issues we do. So we can do computational biology when we do the sequencing of various strains of virus that come in and that you want to make a vaccine for. In fact, I think in his opening statement, Dr. Jernigan had mentioned the fact that the capability both of the CDC and the NIH to do mass sequencing of a variety of quasi-species of any virus, including influenza, relies on computational biology to be able to get to the next step in developing a vaccine.
Is the symmetry pattern of this nanoparticle significant in any way?
Yeah, I mean, actually what it is that the display of multiple components of that stem create the ability to engage what we call the Bcell repertoire of the immune system, so that the chances of it hitting the B cells that will ultimately respond to give you the kind of an antibody response you want, that's a highly immunogenic approach. And nanoparticle approaches to any vaccine is really the wave of the future. And that's what we're trying to do to get away from the situation of having to grow a complete virus and use that as the vaccine the way we're doing in eggs. Here, you use recombinant DNA technology, and you show the immune system only that part of the virus that you want it to respond to, and you avoid all of the other distracting immune responses. That's why the scientific community is so excited about those new technologies.
Thank you. Dr. Jernigan, following up on Dr. Barrett's question, if we find ourselves in a pandemic outbreak, how quickly, with existing technology, can vaccines be produced to catch up with the outbreak?
An example, I think, is in 2017 when there was the identification of a very bad H7N9 influenza virus that started to circulate among poultry in China. Ended up having over almost 2,000 human cases that were exposed to them. We were able to receive the the virus sequence directly from colleagues in China. And with that, we were able to use reverse genetics, like I mentioned before, to actually build the vaccine virus. CDC has the capability to do that under good laboratory practices conditions at CDC, and then be able to hand that vaccine virus to the manufacturers. We can do that very quickly within a matter of days to weeks. However, once we hand it off to the manufacturers, they are bound by the existing manufacturing capabilities that they have. About 18% of all manufacturing right now is in non-egg-based manufacturing. The rest is egg-based manufacturing, which takes at least 6 months. And so getting things to be quicker is going to be an important national security thing for us to be able to respond more quickly.
Thank you. Can you address the autoimmune reaction to influenza vaccines, and forgive my pronunciation, such as Green Beret syndrome?
I'll let you do that if you'd like.
So there has been a rare association of cross-reactivity between some of the antigenic components of a vaccine and certain tissues in the body. There was, again, and this has not been clearly proven yet, but in one of the vaccines that were available for the H1N1 flu of 2009, there was the suggestion that one of the peptides that's associated, which is part of a protein, that was associated with the vaccine induced a response that cross-react with a substance. I hate to use these big words for you. We use a substance called hypocretin, which is one of the neuropeptides that's involved in narcolepsy. So the autoimmune phenomenon of that has been discussed, disputed, but not really definitively proven. So what it is that when you expose the body to a protein, it recognizes it as something that's similar to what's in your body and makes an autoimmune response against it.
Well, my son had a pretty scary reaction to his second DPT injection. Can you speak to that? It was a seizure that was pretty scary, maybe not dangerous, but scared the hell out of us.
Certainly, I mean, febrile seizures is a known reaction just to a number of different vaccines, and I don't know the particulars, but that is something that is possible.
Is it dangerous?
No, for the most part, it's something that does not have a lasting impact.
Okay, thank you. I yield back.
Recognize Mr. Barrett.
Thank you, Mr. Chairman, and we appreciate you witnesses being here and sharing your expertise. So my first question, Dr. Jernigan, deals with, in your testimony, you mentioned the development of a mobile mini lab, a cloud-based platform that can be set in a remote resource-limited settings to process test virus specimens and to send that genomic data up to the cloud for further analysis and action. So could you elaborate on how this cloud-based platform would allow public health officials to address outbreaks quicker and more effectively in a largely rural area like my 4th Congressional District in Indiana?
So yes, I think we were referring to the use of these micro technologies like this one here, which actually, actually is a sequencer. And so you actually take the specimen, prepare it in some little boxes that we take that fit into a carry-on on a plane. You prepare them, and then you just simply inject it in here. There's a way that you can actually do what's called barcoding of the specimens and do multiple specimens at one time. And with that, you get a sequence. And the sequence just tells you the genes of the influenza viruses. So this is something that we have demonstrated in various different settings. We actually did take it to Iowa, to a swine fair, where we actually swabbed a number of the show pigs. And at that, we're able to quickly determine if they had influenza, the swine influenza that was circulating among that group. That data plugs into a laptop through this little USB port. And then on the laptop, it runs a lot of the information and prepares the signal that gets sent up to the cloud, where we have a process called IRMA. IRMA is a tool, a pipeline tool that actually takes the data and uses machine learning and artificial intelligence to try and determine which of the flu viruses are actually in the sequences. That information then gets pulled down by our bioinformatics staff at CDC, where they can then, if needed, generate a vaccine virus. And so this allows us to take the tool to the place where the problem is occurring, rather than having to try and figure out how to get viruses to the CDC.
So to take that one step farther, you could regionalize, or wherever you collected your data, then you could develop a vaccine specific for that area.
It's possible.
Quickly,
The manufacturing process would let you probably not be able to do that, but yes, you can tailor what you know about in certain regions. I think Dr. Watkins will probably get into some of the data issues in the subsequent testimony.
So you mentioned pigs, and I have a background in agriculture, so when you were swabbing those pigs, any thoughts on the African swine fever? I think
Yeah, so African swine fever is something that's different than the swine influenza, and so I'm not an expert in the swine fever, but certainly these same kinds of technologies could be used anywhere in the world to do that kind of detection.
Thank you. Dr. Fauci, you have any thoughts on that area?
Yeah, the point that Dr. Jernigan made is interesting. I'm in some respects glad you brought that up because we constantly get people confused between African swine fever and influenza that's in pigs that could recombine with an influenza to give us a pandemic. It has absolutely nothing to do with that, but sometimes people get confused when they hear the word African swine fever, which is really completely unrelated to influenza.
And I appreciate that. That's part of the reason I mentioned that. So I thank you. I yield back.
Thank you. Let me recognize Mr. Foster.
Thank you, Mr. Chairman, and thank you to our witnesses. Let's see, back to the nanoparticle universal influenza. Can you, I guess, Dr. Fauci, say a little bit about the nature of the nanoparticle and how you actually bond the stem sections to the nanoparticle?
Yeah, it's very interesting. It's the beauty of nature. It's a self-assembling ferritin particle. It's the ferritin protein from a bacteria. And what it does is that when you combine the genes of both, when they express themselves as the nanoparticle, which symmetrically has the stem
Bonding
of the
site.
hemoglobin.
So they just fit properly.
They just fit properly.
Okay.
You know, it's I hate to use this word, but it's almost like a miracle of the natural selection
So
of
the
coming out.
nanoparticle is actually just a larger protein.
Exactly.
Folded in a
Precisely.
specific geometry.
Right.
Okay, and now, if I was reading your slides correctly, the stem section is highly preserved but not absolutely preserved.
Right.
And so are you then going to need several versions of this, or are there dozens of versions, or just in terms of the stem variability?
We don't know, but we believe that we will not need very much because even though it's not completely preserved, we don't believe that the mutations that occur in the stem have a functional relevance in making it different from one to the other. So everything we've done so far, where we've looked at the stem, and we just recently completed a series of experiments where you made antibody against multiple components of the stem, and then you use those antibodies to screen the entire group of the group 1, which contains 10 of those H's, and it just neutralized every one of them. So we think, not 100% sure, that if we get a series of antibodies against multiple components of the stem, we could probably knock out an entire group. And there are 2 major groups. So I think we're going to need at least 2, but I don't think we're going to need 10.
Okay, that's fascinating. And you mentioned this is in phase 1 clinical trials at NIAID Vaccine Research Center? Which
Yes.
That's human safety?
Yes.
And is it proven effective in animals?
Yeah.
Okay.
Yeah.
And so it's all the way through safety and effectiveness in animals, and is it safety in humans?
Right. What we showed in animals is that when you injected it into the animal, you got a complete array of antibodies against the whole panel of the flu. You don't challenge them with every single one, but you know you have a protective level of antibody.
Fascinating. Okay, I'm changing the subject a little bit. Dr. Jernigan, can you say a little bit about the unique challenge of achieving high rates of immunization in immigrant populations, where they very often have a lot of reticence to connect to anything official because of the demonization of immigrant communities?
Relative to my earlier comments about ways to protect the community as a form of increasing vaccine confidence, certainly there are communities that don't value the vaccine, and so I think the better way to get at those groups is to really identify what are the factors that are leading them not to get vaccinated.
Yeah, well, in the case of immigrant communities, you know, frankly, following the 2016 election, I talked to principals in minority communities in my district who were turning kids away from school because they were not being immunized, because they were terrified that ICE was going to come get them if they got their kids immunized. And these are kids that are U.S. citizens, but they have someone in their family who might be undocumented. And is that something you see? Do you monitor the rates of non-immunization in different populations, and do you see an effect?
I don't know if we have that information. We do look at immunization coverage and look at it by race and ethnicity, but in terms of the specifics around immigrant communities, I don't know that we have that information.
Okay, yeah, if you could do a little
can get
digging
back
and
with you
get
on
back
that,
to
yeah.
us, I'd appreciate it. Let's see, finally, you'd mentioned that it was the meat industry in various forms that was a major player in spreading pandemics and having the viruses. Now, in a world where you had artificial vegetable-based meat, which is one that a lot of people dream about, is that something where you'd be intrinsically less prone to pandemics?
So influenza viruses are in reservoirs, and so humans are one of those reservoirs, and there's human-specific influenzas that circulate among humans. The biggest reservoir is among birds, and the biggest reservoir among birds is migratory waterfowl, and so ducks and geese.
Okay, so a word without migratory.
yeah,
That's
so-
not something anyone really wants.
That would be very difficult to try and get rid of, yeah.
Okay, thank you.
Would the gentleman yield?
Oh, absolutely. I will yield my negative 2 seconds. And
That's wonderful. One of the great challenges those of us in the agriculture industry deal with are migratory birds and migratory animals who move around from Canada to Central and South America. They are the thing that we're most frightened about because in their overflights, they deposit little presents as they go along,
well,
which then are subject to consumption by other forms of livestock that have similar characteristics to the rest of us. So that's an issue that causes us great angst, not, maybe that's just the best place to leave it.
Great. Let me recognize Mr. Gonzalez.
Thank you. Thank you for calling this hearing, and thank you to our panel for all your work. I'm a somewhat new father, 19-month-old son, and obviously the flu with respect to our children is something that's near and dear to my heart and many hearts in this room and across the country. According to a Wall Street Journal article, CDC estimated that over 27,000 children ages 4 and younger were hospitalized with the virus, and 118 died in the 2017 to 2018 flu season. Clearly, these are troubling for any parent. I think the uncertainty may be more than anything. And while immunization levels in the U.S. are relatively high, gaps still do exist, and providers can do more to increase immunization rates among their patients and their colleagues. According to the CDC, fewer than 70% of healthcare providers receive the influenza vaccine each year. How does the CDC engage with healthcare providers to promote vaccination?
So certainly, through a number of different studies, CDC has identified that the one way to get patients vaccinated is to make sure that the healthcare providers are promoting the vaccine as well. If you look at the coverage among healthcare providers, it falls into different kind of categories. The more you are at an academic hospital, the more likely you're to be vaccinated as a healthcare provider. The more training you have, physicians have upwards of 90%. The farther you get away from a hospital and the lower the training, like an aide at a long-term care facility, those are the ones that are not being vaccinated. And we clearly want to get the message out that those folks really need to get vaccinated.
Great. And then additionally, in the last decade, it's projected that fewer than 50% of Americans actually get the shot. What research has been done, or are you all doing, just to get a sense of why folks aren't actually getting vaccinated?
So,
trying to identify root causes here.
Yeah, so there are periodically focus group testing that gets done on different groups to try and find out what the reasons are. The main reason that we've identified in the last few years is that the effectiveness of the vaccine. People don't think it's as effective as it should be, and that's keeping them from getting vaccine. We know now that there are more places to get vaccine than ever, so access is one of those things that may have been a problem, but certainly we're getting over with now.
Okay, and then NIAID has prioritized the development of universal influenza vaccines and has highlighted its research strategy towards this goal in the strategic plan for a universal influenza vaccine. In your testimony, you highlight that one of the main challenges facing the goal of producing universal vaccines is improving vaccine production strategies. Could you tell us about plans to address this challenge and keep working towards a universal vaccine?
Yeah, thank you for that question, Mr. Gonzalez. Yeah, that was the point I was trying to make, that we really need to switch into different, what I call vaccine platforms. In other words, not to require to having to decide on a strain in February and then take 6.5 to 7 months to get it grown and processed to be able to put in a vaccine. Whereas if you do the kind of platforms such as the nanoparticle, which is one of several platforms. So as part of our strategic plan that I articulated in that document that you mentioned, is to try and develop and perfect various platforms so that we can get away from the burden of having to grow the virus.
Thank you, and I will yield my remaining time.
Let me recognize Ms. Stevens.
Thank you so much for this insightful panel, and thank you, Dr. Barrett, as well, for bringing us all here together. We heard a little bit today that despite strong efforts in both the public and private sector, that a universal flu vaccine remains elusive. What scientific advances do you see on the horizon to improve the flu vaccine?
Yeah, I believe the scientific advances will be what I was showing on one of the slides of ultimately being able to develop a vaccine that would induce a response that would have broader coverage. You know, I was just actually speaking to one of the scientists who made a breakthrough discovery yesterday when he visited the NIH, Dr. Ian Wilson from the Scripps Clinic. And in 2009, he developed an antibody from a person who was infected with flu, and it bound very, very clearly to a particular component of the stem antibody, which was interesting. And then he found out that not only did it neutralize the virus that the person was infected with, it neutralized all of the viruses in that particular group, which is the group 1, 10 viruses. That was the scientific breakthrough that allowed us to go to the next step of a universal flu vaccine. So it's breakthroughs like that that I predict over the next few years will make it easier and easier to get to the ultimate goal of a universal flu vaccine.
Now, Dr. Jernigan, did you have anything?
Yeah, I think in terms of the near-term kinds of things, I think what we've been looking at, the main problem in the influenza vaccine right now is one of the virus components. We can only put 4 different components in the vaccine, and one of them called H3N2. That's the problem child of the vaccine. And so that one, we know that when you put it into eggs to manufacture, which is 85% of all manufacturing, it ends up changing that influenza virus so that it no longer looks as much like the circulating viruses that are infecting people. So the use of the egg-based manufacturing is introducing some changes that may be having an effect on the effectiveness of the vaccine itself. So moving to cell-based vaccines, moving to recombinant vaccines may be quicker and may actually make the vaccine to be looking more like the H3N2 viruses that are actually circulating.
Can the federal government play a role, particularly in terms of the tools that are being developed to monitor the effectiveness and safety of our vaccines?
Absolutely. I think at CDC we have a vaccine effectiveness network that we manage. And that one we've been able to expand some, but I think expanding that much greater would allow us to be able to get information about how the vaccine is working better or worse in certain age groups, certain parts of the country, certain types of individuals. So it would give us a lot more information to know how to make the current vaccines better.
Yeah. And then in your testimony, Dr. Watkins, you mentioned that public health data infrastructure is a little outdated and it hinders our ability to prevent outbreaks before they occur. It hinders our ability to respond rapidly when they do occur, and it also hinders, our overall abilities around surveillance data. Could you just speak a little bit about or tell us a little bit about the and Watkins isn't here, sorry. I'm so eager for Watkins, and you're both looking at me like Watkins isn't here, but one of you could talk about data infrastructure, and we will also pay note to Watkins when they arrive.
Yeah. So I think that over time we have seen that there's been an improvement in the use of data at healthcare facilities through electronic health records, et cetera. But the public health establishment has to receive information from multiple different sources. And right now there's not a really standardized or common way that information can come in. Plus it's hard for a state health department to be able to quickly get the information they need to know, is this a case of whatever particular reportable disease? Do I need to intervene quickly? Is this person been vaccinated? From a flu perspective, we currently get real-time information about ILI, influenza-like illness, from a number of different sources, but only about half of that is real-time. The other is doctors filling out forms and things. If we were able to get real-time information from all of those providers regularly, we would be able to know exactly what's happening with flu at a much more local level, more precise data, more actionable data for decision-making.
Thank you, Dr. Jernigan. And yes, it is the race for information and data in this modern age. Thank you, Mr. Chairman. I yield back the remainder of my time.
Thank you. Let me recognize Dr. Babin.
Thank you, Dr. Chairman. Appreciate you. And thank you, two gentlemen, for being here with your expert testimony. Just wanted to ask you, Dr. Jernigan, first, what are some of the emerging technologies and practices being developed to identify different pathogens, targets, and modernize the delivery of vaccines? And pardon me if you've already answered questions like this, but I have a markup on a different floor in this same building, so I just came in. I'm a dentist, and one of my colleagues down here asked me if there were vaccines to eliminate cavities, and that would I be against those. He said that in jest, of course, but we encourage Halloween and things like that okay.
Yeah, so with regard to the diagnostics, I'm not going to address the cavity issue, but in terms of diagnostics, so CDC currently maintains a thing called the International Reagent Resource, which is an online storefront that all of the public health departments in the United States and 143 laboratories around the globe are able to go on and order standard reagents that CDC makes, so that we know that the globe is actually doing the same kind of testing for influenza, so that we can use that information quickly. That uses a process called PCR, or polymerase chain reaction, which is a common way. We're currently updating that to get to some newer kinds of PCR devices. But what's really been game-changing is the ability of genomic sequencing.
Right.
And so CDC has established 3 national influenza reference centers at 3 public health labs in the United States, where they do all of that genomic testing so that we can pick up emerging antiviral resistance, viruses that might be a pandemic virus that's emerging, those kinds of things, so that we can act more quickly.
Thank you very much. That's very fascinating. And what are the main scientific and technological hurdles that stand in the way of the development of a universal influenza vaccine? I caught the tail end of somebody's question, had a similar one like that. And how are you working to overcome these? Dr. Jernigan, if you would. I'm going to ask him one here in just a second.
Well, certainly, I'll let Dr. Fauci talk about all the various different hurdles that are out there. For us, the influenza virus has been able to evade human immunity forever. And so you can get influenza every year. So the task we have at hand is a very difficult one, in that the virus itself the body itself is not able to have long-lasting immunity. So we're trying to find something that the body itself is not very good at.
All right, and Dr. Fauci, if you would just go ahead and elaborate on that as well then.
Yeah, well, there's one hurdle that I think is really a serious hurdle. Even if we get a universal vaccine that would induce a response against a wide array of influenzas, and that is a phenomenon that's really very interesting. It's called imprinting. And what it is that your body tends to make a response against the first influenza or the first antigen that it was exposed to when you were a youngster. So that even later on in life, when you get exposed to that organism, that microorganism again, from an evolutionary standpoint, that was a good thing, because that means that your immune system is primed so that if you see that microbe again, you'll make a really good response. That's great for something like measles or mumps or rubella, which doesn't change. It stays the same. With influenza, it works against you. Yeah. So that what you will do is that if the first, I'm an H1N1 person, in the sense that I was born at a time when H1N1 was around. So my immune system is primed to make a response against H1N1. So if I get exposed to an H3N2 or even get vaccinated with that, even though I'll make a reasonable response, my body will revert to wanting to make a response to H1N1. It's referred to sometimes as original antigenic sin. So the real problem is, how do you get around that so that you can vaccinate somebody and overcome that tendency to make a response against something that you were originally exposed to? That's going to be an important obstacle.
Well, and that was the question I was saving for you, and you've actually mostly answered it, because this is why measles, mumps, and rubella vaccines have a 97% effectiveness, where influenza is only, what is it, 10%?
No,
up
no,
to
that
60?
was a very bad year.
Up to 60% though, right? 10 to 60%.
40 to 60% is
Yeah.
what it is, yeah.
So that's the biggest hurdle we have.
Exactly.
Yeah.
You hit the nail on the head,
All right.
exactly.
Thank you very much, and I yield back.
Let me recognize Mr. Caston.
Thank you, Mr. Chair. Thank you both so much. I am just totally intrigued by this universal vaccine idea, and I want to start, if you'll just humor me as a biology nerd. I want to just follow on Congressman Foster's question. So the fact that the stem has been so preserved, how confident are you that that's because there is something fundamentally that the bug just can't change that protein, versus the fact that statistically the antigens were on the surface, and so as we start developing antibodies to go after the stem, are you confident that the stem won't start evolving into something else?
You know, it could evolve under immunological pressure, but from the standpoint of conserved components, we call them epitopes, parts of proteins. When something is conserved throughout evolution, it's usually because it's critical for that particular thing to survive, whether it's a species, an animal, or a protein. So there must be something about that stem that's absolutely critical to the function of the virus. So we think it's not going to change, but we better be careful. We don't want to make an assumption that is going to turn out to be wrong.
And have the animal studies been of a long enough duration to give you some confidence that there is no, I forget what the word you used was, that immunological?
Yeah, no, to be honest with you, no. We haven't done it for a decade and shown that over a period of time, if you keep vaccinating an animal and making a response against stem, and then years later it's going to evolve, we haven't proven that yet. So, I mean, obviously it needs to be done.
So what, if anything, can we do to accelerate, we on this side of the room, to accelerate the development of these universal vaccines? Is it just the time to get through phase 2 trials at this point, or is there something more that you need?
No, actually, what it is that we, thankfully, have gotten very good support from the Congress to do the kind of work that we're doing for the universal flu vaccine. In fact, in our last appropriation, there was a set-aside that was put in order to stimulate the research in that area. So we are very appreciative of the Congress for what you already are doing.
Okay. I want to pivot, and this is I'm going to take a chance here just because I get the sense, Dr. Fauci, that you and I may share a sense of humor. You know what you call alternative medicine when it works? Medicine. I raise that because we are in a moment where there's this rise in anti-scientific thinking, from climate science denial to the anti-vax movement to, I think the New Yorker last week had this article about the rise in people who think that where the stars were when they were born has an impact on their future. As you think about the concerns to public health, there's one set of concerns that is, the anti-vax movement, people consciously choosing not to take proven medicine. There's a separate risk of people who are consciously choosing to take bogus medicine. Which of those, and maybe I'm phrasing this wrongly, but are those comparable concerns, and are we doing enough to combat both?
I think they are comparable. I think there's danger in both of those. I think you brought up two very important points. There really is an obvious concern about people who are anti-science and don't want to believe the clear-cut science facts. And there is a danger to actually having deleterious effects of assuming the efficacy of things that are bogus and going ahead and doing that. We have, several years ago, established first a center and now an institute for an alternative and complementary medicine to be able to look at some of these things that society and people in the community are convinced works, to prove whether they either do or do not work. So we are doing something about trying to put some scientific rigor to some of these things that are potentially bogus. So that's what we're trying to do in that end. On the anti-science end, the only thing that we can do is to continue to do with Dr. Jernigan and his colleagues at the CDC and what we do at the NIH is to continue to try and get out the message and the evidence-based proof of what works. There's nothing like evidence to be able to convince someone that something works, and you have to keep coming in with evidence over and over again.
So are we doing enough to keep bogus science off the shelves? Because when I go to Walmart and I look down the flu vaccine or the flu medicine, there's some homeopathy up there as well, and I don't know that the average person knows the difference. So should we be doing more to make sure
I think
that
as a society we should be. I'm not sure there's much that we at the NIH or that what Dan at the CDC can do, but clearly there's stuff out there that really doesn't really do anything except potentially harm people.
Thank you. I yield back my time.
Let me recognize Mr. Murphy.
Thank you, Mr. Chairman. I just want to say thank you, actually, professionally to both of you gentlemen. Dr. Fauci, I followed your career since the early 80s when you made such fantastic and landmark discoveries with HIV, and it's really put forward something today now that's manageable. So thank you from our community. Being the last one to speak, I always have to figure out which questions that folks have already asked. But let me go back to one of the things. My colleague pointed out the anti-scientific movement these days, and I actually think that's a major problem. I saw last week that people are now starting back on the flat Earth agenda. And I want to go back to the anti-vaccine movement that's going back in our country. I wonder if you could really speak to that, what it's done as far as populations at risk, and where do you see that going in the future? Because it is a major issue these days.
Well, certainly, I think there are pockets where individuals are talking with one another, some schools, that kind of a setting where folks are actually hearing from each other rather than looking to see what the science base is or listening to physicians. And so those pockets, I think, can lead to more and more children, for instance, not getting vaccinated for school, to get into school. I think it's important for us also to recognize that people get their information in multiple different ways now. And so for us to be nimble on how it is that we get the science-based information, the evidence-based information to those folks, identify what their needs are, and then provide them the information that they need. But until you address those specific groups, I think, with information that is valid to them, I think it's going to be actually very difficult.
Thank you. One other issue I'd like us to revisit is Ebola. I don't think people in the United States really understood the gravity of what would have happened if that had gotten into Lagos or any of the other places in the future. And I was wondering if you could talk a little bit more just about the vaccine with Ebola. Does it mutate on the level that the other ones do? And can you, just for edification, just explain to folks the infectivity rate of the Ebola virus versus the HIV virus, for example? I know it's on a multitude scale more infective, but I think giving an example would be helpful.
Well, Ebola, unlike influenza, which drifts and mutates, is pretty stable. It's an RNA virus, so there's always mutations, but the mutations have not proven to be functionally relevant. So if you do a sequence of Ebola in a strain in West Africa, which was Ebola Zaire, the Ebola that's now in DRC is still Ebola Zaire. There are different types of Ebola. There's Ebola Sudan, Ebola Zaire, and others. But within Ebola Zaire, which is the one we're dealing with right now, it really has not been a problem that it has mutated to the point of being functionally relevant. So you can measure point mutations, but they don't change anything about it. I think the question you asked is, what is the relationship with the vaccine? The relationship with the vaccine is that the vaccine has worked, and any change in the virus has not had any impact on the vaccine. So it looks pretty good. So we're, as I mentioned a little bit earlier in the testimony, we've now distributed over 250,000 vaccinations in the outbreak in the DRC. Second part of your question is the issue of how it's transmitted. In an untreated, unvaccinated arena, such as what's going on in the DRC right now, that is, the mortality of that is about 70%. It's transmitted only by direct contact with a contaminated bodily fluid. Right. And that was really important. So if someone gets Ebola and they're incubating it and they get a fever, but they're not having diarrhea, they're not having bleeding, they're not vomiting, that person is really quite non-contagious. And that's the reason why there wasn't a concern of people back when the patients in Texas got infected. There was a concern that those 2 nurses were infecting people, and they were not. Whereas when you get something like influenza, influenza is transmitted by the respiratory route. And there's a window of when you're actually not really very sick, when you can actually transmit it because you're shedding virus for a period of time before you get sick and after. So there really is a rather substantial difference in transmissibility. It is tough to get infected with Ebola unless you have direct contact with a really sick person, whereas you can get influenza on an elevator when the person next to you sneezes. So that's a big difference.
Thank you. Thank you, Mr. Chairman. I'm going to yield back my remainder time to Mr. Posey.
I thank the gentleman for yielding. Mr. Chairman, I'd like to add one more document to the documents. It clearly indicates that while these vaccinations are safe for most people, there are some for whom it's not safe. The Vaccine Injury Trust Fund has paid out over $4 billion with a B, which they did not mention. 46% of those were for influenza-based vaccinations. So didn't want to ruin the love in here, but I think we should not be cavalier about those for whom it's inappropriate, and that we do try and identify who it might not be appropriate to receive those shots for public safety in the future. Thank you very much.
Great. Let me recognize Ms. Bonamici.
Thank you, Dr. Chairman, and thank you, Dr. Fauci, for reminding us that we can get flu in elevators, which we ride in all the time in this building. I'm really glad I got my flu shot. And thank you to the witnesses for being here today. You know, when we reflect over what happened last century, we made such astounding success developing vaccines to eradicate pernicious diseases. In the United States, we've essentially eliminated polio and smallpox and diphtheria, and in the rest of the world, largely defeated those. The World Health Organization estimates that vaccines have prevented at least 10 million deaths between 2010 and 2015. That's pretty remarkable. But in this hearing today, we're acknowledging that there's still a great deal of work to do, especially with influenza, one of the most pervasive infectious diseases globally. Yet, despite all the efforts, we're still struggling to effectively predict or respond to those annual epidemics because of the rapidly changing nature of the flu, as you both discussed. The good news, as our witnesses indicated, is this exciting cutting-edge research that's being conducted throughout the country to develop new approaches. Thank you, Dr. Fauci, for bringing your model. Thank you, Dr. Jernigan, for bringing your mobile lab. And a lot of that work is federally funded or supported, which is why I'm glad we're having this hearing today. Some of that innovative research is happening at the Oregon Health Sciences University in Portland. Dr. Jonas Sacha and his team are working on a novel method of long-term flu vaccination that inserts pieces of target pathogens into cytomegalovirus, or CMV, to trigger a response by the immune system's T cells when the body encounters flu virus. I don't understand what that means, and I'm hoping you will explain it. Dr. Fauci, are you familiar with this approach? Can you briefly explain how it functionally differs from the one you described in your testimony, or more traditional efforts that rely on antibodies, as well as comment on the importance of pursuing varied methods in search of a breakthrough? And Dr. Jernigan, if you're familiar as well.
Right. So the person you're referring to is named Dr. Louis Picker, and he has established a vaccine platform which uses a cytomegalovirus, which is highly immunogenic. And what that platform is, it's called a vector platform. So he takes a virus that we know and have experience with, cytomegalovirus. He inserts into the virus the gene of a particular protein that he wants to make. He's done it with tuberculosis, he's done it with HIV, and he's doing with other pathogens. So what happens is that if you wanted to make a vaccine, which he's trying to do against HIV, he takes the gene that codes for the outer protein of the HIV called the envelope, he sticks it into the cytomegalovirus, and he injects it first into an animal. He hasn't done it into a human yet course, there are some safety issues there. Cytomegalovirus is not a benign virus, so it needs to get a big scrutiny from the FDA. But in the animals,
Interesting, huh.
it's been very effective. He injects it into the animals, it starts to replicate, and it starts pumping out this protein, which is the HIV protein, and he's created, in the animals at least, a pretty good HIV vaccine.
Fascinating. The project I was mentioning was at the Oregon Health Sciences University Vaccine and Gene Therapy Institute.
Correct.
In Oregon. Also, I wanted to ask about the, Dr. Jernigan, about the flu site website. Since 2013, the CDC has engaged in efforts to use this predictive data analytics. How's that working? And the public-facing website, what are you learning from that?
Yeah, so this is a network where we have over 25 different academic modelers. These are individuals that use various different sources of information, social media, weather, all kinds of different information. We provide them some inputs each week, and then they have to tell us what they think is going to happen in terms of, is the flu going to peak this week? When's it going to start, et cetera? So it's a way that we are trying to get not what's happening with flu now, but what is flu going to do. We think that's important so that when we have a pandemic, we can use that information to inform folks. But during regular seasons, that information can be quite helpful for an outpatient clinic, knowing when they need to increase the amount of staff, for a hospital, knowing if they need to have more beds in the ICU, even for pharmacies to know when they move things around.
Some places run out of flu vaccines. Dr. Jernigan, data from more than 100 countries is used to determine which viruses, influenza viruses, are recommended for inclusion in the annual vaccine. What challenges exist for collaborating with so many countries to share data and make sure that that's usable, and what, by everyone, what can be done to improve the international disease surveillance and data sharing so that we can better prepare?
Right, so in the United States, we have very good view of what's happening with influenza, with thousands of viruses that we characterize here. We work with 143 other laboratories, receive viruses from them, but there are blank spots on the globe where we don't know what's going on. So the more we can get improved surveillance, better genomic surveillance in that setting, more timely information from them, that helps that country, but it also helps the rest of us to know what's going on with flu, know if pandemics are showing up, and to make better vaccines.
Thank you. I see my time has expired. I yield back.
Thank you. Let me recognize Ms. Wexton.
Thank you, Mr. Chairman, and thank you, Dr. Jernigan and Dr. Fauci, for joining us here today. In October, the CDC released some new statistics about maternal vaccinations. And I was kind of surprised to see that only 1 in 3 pregnant women receive both flu and whooping cough vaccines, because women with the flu are more than twice as likely to be hospitalized if they're pregnant, and nearly 70% of whooping cough deaths occur in children who are younger than 2 months of age. However, flu vaccinations during pregnancy reduce hospitalization of babies less than 6 months old by an average of 72%, and whooping cough vaccinations will lower the hospitalization of babies by 91%. I think we can agree, I hope we can agree, that that vaccinations are a critical part of prenatal care for expectant mothers. And I understand that, that Dr. Jernigan, you mentioned in your testimony that fewer than half of adults in the United States will get their flu shot because they have a perception that the flu vaccine is not effective. And I know that you've already talked a little bit about misperceptions and false information that's out there, but how can we more effectively communicate the benefits of flu vaccine?
So with regard to pregnant women, I think it's currently around half are getting vaccinated for flu. And so that actually is that's a real success story. Over the last several years, we've seen it really rise to that level. Clearly, we need to do more, and clearly need to do more with the other vaccines that are for pregnant women. If you look at who's getting vaccinated, while only half of Americans are getting vaccinated, you can actually see that the most vaccinations are happening among the old and the very young. And so trying to get at those groups that are late in their teens, 18 to 49 years of age, that's the group that we really need to get at to start increasing the amount of vaccinations. So that's going to take targeted efforts, really using social media and other approaches to get to them.
And just get them used to getting a vaccine every year. Just make it an annual thing.
Part of the problem is that you have to give a vaccine to flu every year. Plus, that's a group of people that probably don't avail themselves of a lot of preventive health care and don't go to the doctor a lot. So I think getting that group in is a challenge, but one that we need to work on.
And I'm glad you brought up social media because, you know, we have seen a lot of how social media can impact lives in a good way or a bad way. And one of the issues with social media is that information spreads so quickly. The viral nature of it allows people to communicate in a bubble without external sources that point out when something is just plain false or something is true and has withstood peer review and all that. So it's something that we've seen across committees in other contexts as well, but here in this issue, we're talking about lives are at stake. And earlier this year, the American Academy of Pediatrics sent letters to the CEOs of major social media platforms, including Google, Facebook, and Pinterest, and highlighted the growing harm to children from vaccine misinformation that spread across their sites. And I understand you have already spoken in some of your testimony about the misinformation and how it spreads. But do you, and this is for both witnesses, do you think that these platforms are doing enough given that lives are on the line?
I certainly think people access their information in multiple different ways than they used to, and so making sure that we get our information that is scientific-based, evidence-based, in the format that is going to be reused, reusable in that setting, I think is an important thing. I don't know if you want to talk about that.
I agree. I think we can do more and we can do better, but I think that the platforms that we have now to get the message out, I think are having some positive effect, but clearly it's a challenge that's not going to go away. We're going to have to keep on it. It's not going to be a problem you'll solve and it's over. We have to keep at it over and over again.
Very good. Thank you so much. I'll yield back.
Well, in closing, just two other questions that come up repeatedly. Folks will say, oh, it's almost the end of November. I've already made it this far. I don't need the flu shot this year. Is that correct?
So our recommendation from the Advisory Committee on Immunization Practices and CDC is that we recommend you get your vaccine, if possible, by the end of October, but as long as influenza virus is circulating, we recommend you get a vaccine. So it is not too late to get a vaccine. Our goal is to try and get people vaccinated prior to the season start so that there's 2 weeks of time before they allow their immune systems to build up so that if they get exposed. But clearly, we recommend that you continue to get vaccinated now.
So since I know most of America is watching this hearing and not another hearing, it is not too late to get your flu shot.
It is not too late to vaccinate.
And then another question that comes up occasionally is nasal flu vaccine versus flu shot. Any recommendations or equally effective?
So currently, CDC does not have any preference for any one vaccine over another. There are personal preferences and parental preferences with regard to the live attenuated influenza vaccine, the nasal vaccine. So there's no preference for one over the other. They're all listed as effective as each other.
Great. Well, I once again want to thank both of you for your service to this country and service to medicine. And again, for those watching at home, flu shots are safe, or vaccines are safe, vaccines are effective, and vaccines save lives. Thank you, and we'll recess for a few moments and allow the second panel to get seated. Thank you.
Great, thanks.
Thank you.
Doctor, nice to be with you.
That's possible. Thank you.
He's public health. He's medical examiner. I got him.
Oh, okay.
Oh. Maybe somebody brought it. Okay, they have it. Okay.
Okay, good. I will thank him on the floor then.
They said they put it in his books.
He's CPA, he's methodical and thorough. Let's get fired up.
You had to bring a bird food, huh?
You bet. Because Dr. Foster, when he depopulates all of the countryside, we'll still have bird poop out there. And we will have the most god-awful giant wildfires. It'd be like volcanoes when you take all those herbivores off the plains.
That's right. Goats are like the most effective
Welcome back. At this time, I would like to introduce our second panel of witnesses. The first witness in our second panel is Dr. Sharon Watkins. Dr. Watkins is the Director for the Bureau of Epidemiology and the State Epidemiologist for the Pennsylvania Department of Health. She is also the President of the Council of State and Territorial Epidemiologists. Dr. Watkins is responsible for management and oversight of the Bureau of Epidemiology, which includes the Division of Infectious Disease, Environmental Health, and Community Health. Dr. Watkins has led disease surveillance and outbreak response efforts, including those related to Zika, healthcare-associated infections, measles, and hepatitis A. Dr. Watkins has over 40 peer-reviewed publications and over 20 years of experience in applied public health and epidemiology. Thank you for being here, Dr. Watkins. Our second witness is Dr. Robin Robinson. Dr. Robinson is currently Vice President of Scientific Affairs for RenovaCare Incorporated, directing development of cellular therapies for wound healing. Previously, he served as the first director of the Biomedical Advanced Research and Development Authority, BARDA, and Deputy Assistant Secretary for Preparedness and Response from 2008 to 2016. He also served as BARDA's Influenza and Emerging Disease Program Director from 2004 to 2008. Dr. Robinson was the recipient of the Department of Defense Clay Dalrymple Award in 2008, the HHS Distinguished Service Award 3 times, and a finalist for the Service to America Medal in 2009. Thank you for being here, Dr. Robinson. As our witnesses should know, you will each have 5 minutes for your spoken testimony. Your written testimony will be included in the record for the hearing. When you have completed your spoken testimony, we will begin with questions. Each member will have 5 minutes to question the panel. We'll start with Dr. Watkins.
Dr. Barrett, Ranking Member Lucas, and members of the committee, thank you for the privilege to appear before you today. My name is Dr. Sharon Watkins, President of the Council of State and Territorial Epidemiologists, CSTE, and State Epidemiologist for the Pennsylvania Department of Health. CSTE is an organization of 56 member states and territories representing applied public health epidemiologists or disease detectives. We work every day in partnership with CDC to detect and respond to influenza outbreaks, gain an understanding of potential changes in the virus, and deliver life-saving vaccines. I have witnessed the devastating impact of seasonal influenza, the 2009 H1N1 pandemic, measles, and many other vaccine-preventable diseases in the communities I serve. Public health threats require efficient, timely responses that rely on a network of public health agencies at all levels of government in coordination with healthcare providers. Response to outbreaks happens at the local level. Data on the age group affected, vaccination status, underlying illness, pregnancy status, and whether the outbreak is in a school or a long-term care facility. For example, they all need to be rapidly identified so that we know where to respond and what is needed. Unfortunately, this public health network is choked by antiquated data systems that rely on obsolete and sluggish data sharing methods. Faxes and phone calls are still in widespread use. The system is in dire need of security upgrades. Lack of interoperability, reporting consistency, and data standards lead to errors in quality and completeness, timeliness, and communication. I have stood before communities in crisis who are justifiably bewildered and angered that public health cannot access disease data or access it faster. How is it that I can simply log into a portal and get my medical test results in a matter of minutes, and you, who are charged with protecting public health, don't have access to today's health data? It shocks people to learn that we do not have national coverage connecting hospital emergency departments with public health surveillance systems. About 40% of all ED visits are not submitted to public health departments, leaving us flat-footed in identifying and responding to severe flu infections among high-risk groups, including pregnant women, children, and the elderly. We are now entering flu season and are challenged by the concurrent outbreak of lung illness associated with e-cigarettes. Public health is urgently deciphering faxed medical records to distinguish e-cigarette-related cases from flu cases. This information arrives piecemeal at different times through different channels. Try to decipher Addendum 1 in my written testimony. It's a 4-page sample of a 350-page fax medical record received by the Pennsylvania Department of Health on one of our e-cigarette cases. Providers already have this data shared and collected in electronic health records, but cannot rapidly share these data with public health, who have no way to receive them electronically. Death certificates are still filed on paper in some states, and only 63% of all death certificates are submitted to CDC for national aggregation within 10 days. Regrettably, most pediatric deaths occur in unvaccinated children, and it takes weeks to uncover and link the flu death with vaccination history, causing lags in communication to stakeholders who need answers to these questions. CSTE and our partners, the Association for Public Health Laboratories, NAPHSIS, and HIMSS, together with more than 90 other institutions, believe the time is now to step up and take a coordinated approach to building a 21st century public health data superhighway. This superhighway will collect health data from healthcare providers and report it automatically to public health departments and link it to other key data, including birth and death records and immunization registries, and share that data seamlessly and securely with CDC. The technology is here. What we really need are resources. That is why the proposed funding of $100 million that was included in the House Labor, Health, and Human Services Appropriation Bill to support data infrastructure at the CDC is urgently needed. During your ongoing deliberations, CST hopes you will consider the need for a modernized electronic, interoperable public health data system and skilled public data health scientists to strengthen public health's best prevention strategy, vaccination. We recognize this effort must be funded with new money rather than cut already underfunded public health. Without federal support, public health surveillance modernization will remain unattainable and the nation will suffer. We look forward to working with you, and I thank you for the opportunity to testify before you today.
Thank you. Dr. Robinson.
Good morning. Thank you, Acting Chairman and Ranking Member Lucas and distinguished members of the committee. Thank you for the opportunity to speak with you today. I'm Dr. Robin Robinson, currently the Vice President of Scientific Affairs at RenovaCare, the former Director of BARDA and the DAS at ASPR, and a developer of influenza vaccines in industry. 4 years ago, I testified at as the BARDA Director before the House on the state of affairs for seasonal influenza during a harsh season and what we could do to remedy mismatched flu vaccines. Since that time, seasonal influenza has returned each year and brought illness and death despite our medicine cabinet full of vaccines and antivirals. New influenza vaccines with adjuvants and a fourth strain of influenza vaccines and a new class of antivirals were added since 2015. Yet we still have not solved the chief issue with influenza vaccines, poor effectiveness. Our domestic capacity to produce pandemic influenza vaccines has quadrupled since 2005, thanks to our investments in new cell and recombinant-based production technologies. However, our ability to manufacture and make available pandemic influenza vaccines are not fast enough to preempt pandemic peak effects. Lastly, many universal influenza vaccine candidates have emerged over the past 40 years, but none have crossed the finish line. Today, I wish to address poor vaccine effectiveness, slow vaccine production, and elusiveness of universal influenza vaccines. Vaccine effectiveness and universal influenza vaccines are both dependent on the selection of viral antigens that can elicit long-lasting, broad, and strong immunoprotective responses across many different influenza virus subtypes. An ideal universal influenza vaccine would elicit strong and lasting immunity against currently circulating and drifted strains of seasonal influenza viruses to obviate the need for annual immunization against seasonal influenza and serve as a vaccine primer for pandemic. The story of universal influenza vaccine development is long and woeful. For the past 40 years, multiple waves of innovation have driven universal influenza vaccine development. One of the earliest and most expensive efforts was by Merck in the 1980s and 90s, focusing on vaccines comprised of the highly conserved influenza M2 matrix protein. However, the M2 vaccine candidates were poorly immunogenic. Next, vaccine candidates targeted the highly Conserved NP and NS2 proteins were developed and shown to be poorly immunogenic as well. The story changed with two discoveries, one of which Dr. Fauci mentioned earlier. Made this decade, antibodies were discovered in 2011 to specific epitopes on the conserved stem portion of the viral hemagglutinin protein and shown to bind and neutralize widely diverse influenza viruses. This discovery has led to a new development wave of chimeric hemagglutinin and hemagglutinin stem vaccine candidates that are undergoing clinical evaluation presently. The other discovery, which occurred this year, was the finding of antibodies to conserved epitopes on the viral neuraminidase protein, which has been a target for antivirals for many years. These antibodies bind and neutralize widely diverse influenza viruses. This discovery will likely initiate another wave of vaccines, as scientists will likely include this specific neuraminidase protein in their next generation of flu vaccine candidates. On the issue of more rapid production of influenza vaccines, new synthetic messenger RNA vaccine technology may expedite vaccine production. These mRNA vaccines do not require the isolation, adaptation, and production of viral vaccine stocks like the current egg and cell-based influenza vaccines. Weeks to months may be saved in vaccine production. This time savings may allow the late production of seasonal influenza vaccine strains when a mismatch occurs between circulating influenza viruses and seasonal influenza vaccines. Similarly, the production time for 600 million doses of pandemic influenza vaccine may be reduced from 6 months to 3 months and become available before the pandemic peaks. As added value of messenger RNA vaccines may be a faster and easier way to distribute and administer these vaccines. Many messenger RNA vaccines are encapsulated in liposomes or nanoparticles, as Dr. Fauci stated, and which may intrinsically have adjuvant properties and the ability to administer vaccines transdermally, hence trading a syringe and needle for a self-administered patch. None of these innovations and discoveries will make it into the influenza vaccines of the 2020s without immediate and sustained multi-year funding and authorities to NIH, BARDA, FDA, and CDC to execute with industry partners the pandemic plans of yesterday and today. Your continued wisdom, generosity, and support have carried us this far. Help us finish the journey. Thank you.
Thank you, Dr. Robinson. At this point, we'll begin our first round of questions. The chair recognizes himself for 5 minutes. Dr. Robinson, thank you for your service at BARDA. I had the chance to meet with the top folks at BARDA, but another international organization that I've also had the chance to meet with is CEPI, and it is a organization that is looking at bringing the international community together, along with the private sector, et cetera, to look at vaccines for emerging diseases and so forth. If you could elaborate a little bit more on the mission of CEPI, and one of the big disappointments for me is that the United States currently doesn't participate in CEPI, and I'd be curious about your opinion as to whether the U.S. should participate, and if you want to elaborate on that. Can you turn your microphone on?
I'm sorry. Thank you for the question. I always smile when CEPI is brought up because my former deputy at BARDA was Richard Hatchett, and he is the current CEO of CEPI. Should the U.S. participate in the activities of CEPI against emerging infectious diseases and development of vaccines? And the answer is that we already are. And this was when the inception of CEPI occurred back around 2014, and it actually became a reality in 2017. And that the NIH and BARDA specifically had investments in emerging infectious disease and specifically on vaccines such as Ebola, Zika, and others. And that was part of our contribution. We would continue as the U.S. government's efforts in these specific areas. So we do actually support what they do. In many cases, we have contracts and grants that actually are supporting the same projects that they're working on, but without duplication of exactly what they're doing.
If we play off of that for a moment, and again, my interest in pandemic preparedness and some of the threats, if we look at emerging diseases and some of those pandemic threats, what is our capacity to, within the private sector to quickly ramp up if we see an emerging pathogen, quickly identify a potential vaccine to mitigate that pathogen? Just from your perspective as an expert in the field.
So I'll give it in the context of when I started my public service in 2004, in which it would take months to years to be able to respond to a new emerging pathogen. My first assignment was on H5N1 avian influenza viruses and how we could actually make a vaccine towards that. Since that time, we actually had a real live test in 2013 with the emergence of H7N1 viruses. What normally would take about 6 months to actually produce those vaccines, we actually brought that down to closer to the 3 months, and there was a specific reason why. First, as you heard from Dr. Fauci and Dr. Jernigan, we were able to get the sequence of that virus immediately, and actually it was on April Fool's Day of 2015, and actually moved forward within weeks to actually have that sequence distributed not only to the vaccine manufacturers of egg and cell-based producers, but also for recombinant products. By the summer, we actually had those vaccines in clinical trials. And so in record time, we were able to do that. Many of the innovations that we are talking about today would even expedite that further. And our goal, of course, is to actually have pandemic vaccine not only produced, but available within 12 weeks.
Great. And Dr. Watkins, in a prior life, I was chief medical officer for Sacramento County, so did a lot of public health work, and it makes me chuckle because we would get information faxed to us, and most of the public wouldn't believe that in this day and age in 2019, a lot of public health records and information is fax-based. So you talk about interoperability, you talk about collecting data and creating big data sets. Could you just elaborate a little bit more on what that would allow you to do in terms of more rapidly identifying potential outbreaks, et cetera, and why a more robust interoperable electronic medical record or an electronic public health record would allow you to do your job better?
Sure. When I think about medical delivery of the healthcare system today, it's amazing the advancements that have been made, but I think public health has been left behind a little bit. And we are still dealing with faxes, and we are still dealing with phone calls and spreadsheets, handwritten spreadsheets, and it really does impact our ability to quickly respond to a situation. So if immunization records were able to be quickly linked to our electronic, to our disease reporting system, if we were able to get electronic case reports and see data as it's coming in and digest that in the health department, we would really be able to respond much faster. Much of what we do in many of the pandemics or the emerging threats that we have today is scratch our heads, and we're really struggling with the data sharing and the data management of so much big data. Public health needs to have our systems renewed and reinvested in. And CST has produced this book in conjunction. There are a lot of stakeholder stories in this that talk about why public health and the time is now to invest money in our data systems.
Great. The chair now recognizes the ranking member, Mr. Lucas, for 5 minutes.
Thank you, Mr. Chair. Dr. Robinson, in your testimony, you highlight that clinical trials have shown that vaccines are stockpiled, remain highly effective even after 10 years in storage. How has BARDA worked with the industry to improve the shelf life of stockpile vaccines and other countermeasures in the event of a pandemic emergency?
Thank you for the question, sir. We started in 2005 building our stockpiles, we stockpiled for pandemic influenza. These would be to treat those individuals that are highly vulnerable, at high risk, and our critical workforce to make sure the country still operates in a severe pandemic. So around 27 million doses. And that was actually for all the different strains that have been shown to have pandemic potential, from the H5N1 viruses, the H7N1 I just described a moment ago, to the new waves of H7N9 viruses. Through the IRAT process that the CDC has with BARDA, FDA, and NIH, we actually meet twice a year, go over these strains to see which ones are available. But in 2015, we said that, and it was a question that actually came up from the members here, is the vaccine that you have stockpiled in these companies, is it still good? And the answer was, well, we know that the potency assays look really good, but we said that's not enough. So we went and actually did a clinical study using newly made H5N1 virus vaccine against vaccine that had been made 10 years before. And the results of that in the BRIGHT study, which have been published, show that they were equal. And they were still highly immunogenic and could be used without or with an adjuvant to protect those individuals.
Thank you, Doctor. Dr. Watkins, you suggest that the use of artificial intelligence or machine learning could be a useful tool to identify outbreaks early and encourage individuals to get vaccinated. Can you elaborate further on how this technology could be utilized?
Sure, thank you for that question. Public health does have a lot of data. It's not interconnected, and I think that the ability to look at birth and death certificates and immunization rates and existing comorbidities and combine that with census tract information and behavior information and information on poverty and immigration status, all of those other data sets helps us better understand at the community level what are the hesitations or what are the limitations to vaccination or access to health care or maybe language barriers. And when we're able to use all the data that Google has at their hands and we don't, I think we're better able to target where efforts should go. As an example, during the opioid crisis, we and other states funded by CDC have been looking at vulnerability assessments. So we're looking at where are our deaths happening due to overdose? Where are babies being born with neonatal abstinence syndrome? Where are rates of hepatitis C and HIV increasing? And where does that overlay with poverty and some other statistics? That's use of big data in a state to really look at vulnerabilities and target where we should be working. We could be doing that with many more things had we the technology and interconnection.
Thank you, Doctor. Yield back.
Let me recognize Dr. Murphy. Oh.
Thank you, Mr. Chairman. Thank you guys for coming this afternoon, and appreciate your expertise. First question I'm going to have is for Dr. Watkins, because I was looking through some of the copies that you have of medical records and everything, and having experienced the explosion of the electronic medical record just in my own practice in the last 25 years, I see the challenges for it. If you could wave a magic wand, there is a way to pull data out of these reports and quantify it. What would it look like? Because I preface it by saying we have so many different medical record systems in our country, most of which don't talk to one another. And unless we have literally a single system, I'm not sure of what this would look like. So I'm just interested in your thoughts about reality of this, how we do this, because I think the purpose is altogether a great one, but the devil's in the details. What does that look like?
Thank you for that question. I would also refer you to this report that has been done, and we can get you a copy of that. But what we're talking about is modernizing systems we already have. So our laboratory system, which is called LIMS, and its ability to rapidly transmit data between us and the provider and CDC and handle those genetic results needs to be modernized and made more interoperable. Our death and birth certificate registries need to be more rapid. I mean, we shouldn't be having paper records of these important documents. Our immunization registry should be interconnected with our other disease reports. And our electronic disease collection system should be able to know if you've gotten influenza and a death certificate comes in, I shouldn't have to wait weeks. I should be able to see that within real time. So looking at being able to bring those in, and CDC is doing a lot of work on electronic case records and modernizing of all of these systems. What we're talking about is bringing all states up to a better level. Some states are really far behind, and some states are behind in some things but not in others. And when I think about a pandemic or the next emerging issue, I mean, we don't want public health to be the weak link in the chain. We want public health to protect your family, my family, and the public's health with the same tools that private medicine has and the same speed. So that's what we're talking about.
Thank you for the question. It's a daunting task. I think it's a good idea. I will tell you, it adds an entirely additional level of just data entry. But then again, that's what we do. We work on data.
We'd like to get out of the data entry. You know, I have some analogies for you, if I may. And to me, it's like, I'm sure we all have private physicians. We have healthcare providers. And, you know, they're not sharing information handwritten on you. They're not walking your lab test results in a spreadsheet. I mean, they're working in a modern world with modern technology and modern informatics. And public health is the front line for pandemics. We should be working with those same speed. It's like building a space probe and forgetting to put in the advanced communication and data sharing aspect of it. And I feel like in this modernization of healthcare, and we're talking about vaccine innovation, we're thinking about all that, but we need to think about modernization of public health data sharing so that we can be the front line of public health and not be the weak link in the chain.
Great, thank you. Because I agree, those are the issues. It's not cancer, it's not other things that you need the connectivity. Just one other quick question, just Dr. Robinson. I was wondering if you could speak to the fact, we've talked a little bit about the vaccines that come primarily from eggs versus the cell-based and the recombinant. Can you speak to really why you don't believe that the technology of the latter really is taking up, or are we making a good progress towards moving away from the egg-based vaccines?
So because of the efforts we had at HHS and primarily through BARDA, we actually made a paradigm shift where we were 100% egg-based to, as Dan Jernigan said today, 85%. Now, how are we going to move to at least having greater adoption of recombinant cell-based where we don't have some of the problems with mismatches? First of all, we have to realize that the influenza vaccine industry is a commodity-driven industry, and that the way that we were able to move the needle to begin with, it was interacting with them as a public-private partnerships. That has to be revived and continue to go forward with these new discoveries to make it worthwhile for them to have a product so they can get out of the egg-based vaccine business. I will say that there's promising progress that companies that are solely egg-based have actually either bought recombinant vaccine candidates and that are actually licensed now, or they are internally developing new influenza vaccine candidates. So we need to expedite that and facilitate it with the continued efforts that we've had with a good formula before.
One just follow-up. Do you think that the recalcitrance to doing that really is regulatory, or is it the economies of the cost?
It's not regulatory. I mean, industry, and I'm now part of that industry, may say, well, we don't want to do that because we have to go through the entire process of getting a new vaccine licensed to the FDA, but that's the normal course of vaccine development. The real problem is, why spend money when we don't have to? Sure. And that's a reality.
Sure. Thank you very much. I'll yield back mine.
Let me recognize Mr. Cohen.
Thank you, Mr. Chair. Dr. Robinson, and you may have just probably been maybe touched on the first panel, but the whole public media, social media, conspiracy theories about vaccinations causing autism, how much of an effect has this had on people getting vaccinated, and how much of an effect of people not getting vaccinated have on public health?
So there's two parts to that question. The first part is how much, what was the effect of the anti-vaccine groups for autism. We fought this battle during the last decade, and I will say that to a great extent, that battle has been won. And so scientific data was actually shown that there's no link between vaccination and autism. The second part of that...
Let me ask you a follow-up on that.
Yeah.
You say it's been won.
I'm going to answer that because we have a new wave of anti-vaccination, and I'm very concerned about this because they don't have as their true agenda vaccination. They could care less whether it works or doesn't work because they have a hidden agenda for other things of anarchy and other things. And the tactics that they're using are ones that cyber terrorists have been using over the past several years, and I'm very alarmed by that because again, the vaccination is not their real issue here.
Well, there are some that I think, for instance, my friend Robert Kennedy Jr., he's a major anti-vaxxer, and he's not for anarchy.
No.
I think his issue was thinking that mercury as a preservative was the cause. Is that correct?
That is one of the platforms that they have espoused.
Has there been studies to show that that is wrong?
So what we have, that was said by Dr. Fauci earlier, one is it's not methyl mercury, it's ethyl mercury that is in some multi-dose vials of some vaccines. I will say that we made a pointed effort in 2008 with influenza vaccines to remove that, and the manufacturers did this without being mandated to do so, and so that there are single-dose syringes without mercury in those vaccines and those are primarily given to children and to pregnant women. And so there has been major progress on this. And as Dr. Jernigan said in his testimony earlier, that CDC and FDA are mounting efforts to be able to minimize that. But again, the amounts that there and the kind of mercury there are not the kind that Mr. Kennedy has been talking about.
Dr. Watkins, do you have any perspective on this as well? Anything you add?
Thank you. I think public health is clearly worried about these sentiments and that we need to do a better job in communicating the efficacy of the vaccine and the benefits that it does in addition to preventing disease. It also lessens the severity and complications, and particularly for those most at risk, so it prevents death and hospitalization. I think, public health thinks a lot about the fact that where do people get their health information and how do they communicate with each other, and we need to do a better job of producing, convincing messages that are shared on different platforms.
How many people do you know, or can you use a round figure, die annually from the flu?
I don't have that figure in my head, but we can get it for you.
Well, Dr. Robinson, you have a clue?
Yes, sir. At the low end, 10,000, upwards to 48,000 a year, sir.
So those people more likely than not, if they had the flu vaccine, and you don't know that some of them might not have gotten the flu vaccine and it had not been that particular strain, but more likely than not, that would have been reduced greatly if all those people had been inoculated.
That's correct.
Yeah. Thank you. I'm a big proponent of vaccinations. My father was a pediatrician. He gave vaccines. In 1954, he gave the Salk vaccine to second-grade students in the test trials. Had a brother that was in second grade. He gave him the Salk vaccine. I was in kindergarten. He brought it home to give to me. And he had second thoughts because it was outside of his charge. Within 2 months, I got polio. Vaccines are good. I yield back the balance of my time.
Thank you. We'll open it up to additional questions from the members, and I'll start by recognizing myself. In my interest in pandemic preparedness, Dr. Watkins, we've been having conversations with companies like Google, and I know Google has been doing some work in identifying particular search words that may pop up that would then allow us to rapidly say, people are searching the term fever or et cetera to try to quickly go into, let's say, a country in Africa or someplace else. Are you familiar with any of those trials and have they been successful, not successful, et cetera?
Well, public health is aware of those kind of crowdsourcing tools that look at GI symptoms or they look at fever, and but we've not been using them in public health. Most jurisdictions, I think some may have. What we are interested in, because we are a system that uses case-based surveillance. I mean, we know your name if you're sick. We're counting you as an individual. But we have expanded a little past that into syndromic surveillance, where we are looking at de-identified emergency department visits and really gaining a lot of information that way. So I can't say whether the Google has been validated through public health methods, that crowdsourcing. I can say that looking at emergency department, just, are you seeing a spike in this, that, or the other, has been incredibly effective, not just in identifying the uptick of flu, but of many other diseases, including being able to identify clusters of illnesses.
Dr. Robinson, would you want to add anything?
No, I think Dr. Watkins said it.
And yeah, I still think it's worth, as we're looking at global health and pandemic preparedness, to continue to work with these technology companies that, because part of rapidly responding and getting ahead of pandemics is quickly saying, hey, let's get someone out there, let's identify what that pathogen is, and let's see if we can't mitigate it at the source. Is that correct?
Absolutely, but with all due respect, I think public health is undersourced and under-resourced in the informatics world. So our ability to really be doing that is contingent on us being able to modernize.
Do public health information systems speak across state lines?
No, not necessarily, no.
Okay, and that's not because of any regulatory issues that we've placed as Congress. That's just under-resourcing, or?
Well, it's both. I mean, Ohio doesn't have the jurisdiction to see that John Smith in Pennsylvania has influenza. It's my jurisdiction. But we could do a better job of sharing not identified data across state lines. And when there is an outbreak and we need to share that information, we do so securely. Okay. But no, for example, in my state, Philadelphia is on a different surveillance system than the state is, and it does really matter. We have to really work hard to share data, and when CDC wants to see statewide data, we have to work with Philadelphia to harmonize it. It's inefficient.
As a public health expert, let me ask another question about vaccination rates, and I guess let me put it this: when I was a child, I got a lot of my vaccines at school, and I'm an internist by training, not a pediatrician, but it's always occurred to me that, for efficiency's sake, especially for multi-dose vaccines, you've got a captive audience at that school. The kids are going there, but the overhead, if you had school-based nurses or public health nurses that were able to go into those schools to vaccinate their kids, it would be more effective, more efficient, and I'd just be curious from your perspective, Dr. Watkins, if that's something that we made a mistake of moving away from.
Well, we certainly do school-based vaccinations in outbreak settings. That's a perfect setting, and we do use that venue. I think school-based nurses are a resource that is shrinking, and so not all schools have access to that. I think that looping schools into immunization and other kinds of issues is always a goal of public health, and I do think that we've done it broader and have shrunk that footprint, yes.
I mean, I understand that there's probably concerns about liability issues and et cetera that have moved us away from that, but just from a pure cost perspective and efficacy perspective, I think those investments in public health nurses or school-based nurses, the overhead and et cetera, and again, the efficiency, particularly with multi-dose vaccines, because you lose a lot of kids. They don't come in for that, a month later for that second vaccine. And again, I believe you could rapidly boost the number of children that are getting vaccinated, if we were to utilize tools like that. And I guess I'd ask one last question with regards to measles, et cetera. Just, I'd be curious from your perspective as a public health professional, how Pennsylvania and others around the country are trying to address the periodic outbreaks. And again,
Sure. I mean, we're exhausted. I'll just be honest. I was just at a conference with New York, and I can't even imagine what they've had to go through to be able to address those thousands of cases. In Pennsylvania, I think we're at 17 cases. What I think you don't realize is that for every case, hundreds of people are likely exposed. And if it's been close contact exposure, if you were infectious with measles right now, everyone in this room and everyone in this room for the 2 hours after you have left it would have been exposed. Public health notifies you. We track you down where we can. We assess your immunity. We work to make sure that not only are you taken care of, but everyone you've exposed is notified and properly treated. Either you're immune or you're not, and if you're not, and we can't get prophylaxis in time, you may be quarantined. There are a lot of steps that go into measles, and it's an enormous resource drain. It's been difficult for New York and for any of us who have had cases of measles.
well, Dr. Watkins, thank you for your work and all those public health professionals. And Dr. Murphy, if you have any additional questions.
Thank you, Mr. Chairman. Again, thank you guys for coming. Dr. Watkins, let me ask you a question just because we're looking at this in one level of the problems that you face with interconnectivity and challenges, by all means. My question is, what have you done in the state of Pennsylvania to talk to the other counties, because public health departments, at least in North Carolina, are run by counties? What have you guys done on a state level to develop interconnectivity? And I just, on a corollary, I did a lot of work in the North Carolina legislature with the opioid epidemic, and we had people on the border of North Carolina going into Virginia getting prescriptions, vice versa. So we worked close by with our state neighbors to develop a system that somebody in Virginia could know if somebody's jumping across the line and getting prescriptions in North Carolina. It's the same thing. It's state interconnectivity, not necessarily a federal push-down approach. When we look at the nation as a whole of pandemics that are going on, by all means we need to know that information. But these tend to be localized. And so what have you guys done on a state level to address this problem?
So let me just say that Pennsylvania is structured differently than North Carolina. I mean, we have 10 county and municipal jurisdictions. We're home rule, Commonwealth. So they are on our same system of disease surveillance, and so we are able to share that. So what happens are lab reports come in, or a report from a physician comes in to the state health office, and we push it to the jurisdiction. Or to the district office. Mostly if you're in a home rule system, if you're in Pittsburgh, for example, Pittsburgh's seeing their own records. But we do collect it all in the same data system. Philadelphia is large, and they're able to have their own and a different system. So we work with them. We work with them both from a disease perspective. We share outbreak information all the time. We work with them from an IT perspective to try to harmonize what we do. And of course, we're always working with our neighbors, whether it be on hepatitis A outbreaks or measles or sharing of, I mean, patients don't have borders. I mean, you can be hospitalized in New Jersey and go into a long-term care facility in Pennsylvania. Happens all the time. So we keep in touch, but we could do it better, faster, and without loss of information or misinformation if we were better electronically suited.
All right, well, let me just follow up then. Are you not electronically suited in these different counties, and why would you not appeal to your state rather than the federal government to make that happen?
So what I'm talking about is the sharing of laboratory information with disease surveillance, and that is happening at the state level, but it's not an easy connection. We've really not invested money in this in a long time. And for example, our immunization record is not connected to our disease surveillance record. And I'm speaking from the national perspective, CSTE. You've asked me a Pennsylvania question, but I could be answering for many states. I don't know if your immunization record in North Carolina is connected to your disease registry. For many states, it's not. So those are the kind of things that would help us get data and respond faster. In a measles exposure situation, who's been immunized? That's a hard question. It shouldn't be a hard question, but it is a hard question. Sure. And we've resorted to actually going to high schools, the old high school, who's stored records, who've looked them up for us because the physician had gone out of practice, or public health is a make it work kind of system, and we just do what we need to do, but we're getting further and further behind.
Let's see. Thank you. And one other quick question, just for Dr. Robinson. In the success that we've seen with the cervical cancer vaccine, with the against the HPV virus, here I am, a physician trying to put myself out of business. Where are we, and where do you see us as far as other malignancy vaccines? I'm going to give you prostate cancer, for example, because I've seen literature for that for 15 years. I just don't see the door being knocked down. So can you just speak to that briefly and what your experience is and thoughts?
Yes. 20 years ago, when I was in industry, we actually worked on a prostate cancer vaccine and a melanoma vaccines. What has driven the oncology vaccine has been supplanted by the monoclonal antibodies that have been developed with great success over the last 15 years. Mm-hmm. So that has somewhat moved the vaccine programs to, especially in companies, to a lesser degree. Some of those vaccines were extremely promising as we and others were evaluating those in the clinic, and I would suspect that once we reach the peak of the monoclonal antibodies for oncology purposes, that we will actually see a resurgence of vaccines for different types of cancer reappear probably in the next decade, in fact.
Thank you.
Great. Before we bring the hearing for a close, I want to thank both of our witnesses for testifying before the committee today. The record will remain open for 2 weeks for additional statements from the members and for any additional questions the committee may ask of the witnesses. The witnesses are excused and the hearing is now adjourned.